Hello everyone, welcome to DiHybrids. I would like you to take a minute and find this note file in Schoology and drop it in a Notability. It's called DiHybrid Intro. It would be best if you split screen with this video and this note file so you can fill it in as we go. Pause the video and go ahead and do that now. Okay, before we dive into DiHybrids, let's do a really quick review. Let's look back at monohybrid crosses. Remember the prefix mono means one. So we're going to look at one gene with two possible alleles. Okay. So in peat plants, yellow seeds are dominant to green seeds. So let's say I'm crossing one plant that's homozygous dominant for this trait to another plant that's homozygous recessive for this trait. Remember, what we're really doing here is trying to predict the outcome of meiosis. So we're looking at what possible gametes can be made by these plants. So this plant can make a gamete with only a big Y. Okay, so I would put this on one side of the Punnett square. And this plant can make a gamete with only a little y. So I'd put that on this side of the Punnett square. And what we're really looking at is the ova, which is like the egg, one of the gametes in a plant cell, and the pollen, which is like the sperm. And these are going to come together in fertilization, and this is my possible offspring for this trait only. Now, you could finish this Punnett square, but all of your offspring would be the same. Okay? So for monohybrids, where you look at one gene or one trait, but notice when I'm doing my Punnett square, each possible gamete only gets one allele, and that's because in meiosis, we cut our number of chromosomes in half. It's reduction division, so we only get one allele for each trait. So now we're going to build on this. Here we're going to look at the first trait of whether or not the pea seed is round or wrinkled. So let's say this pea right here is homozygous dominant for round. And this pea right here is homozygous recessive, which means it's wrinkled. Round is dominant to wrinkled. Now when we start to consider the type of gametes these peas could make, this pea could only add could only add the dominant R to its gamete. It would only put in the big R. And this P would only put in the little r. Okay? So Mendel wondered, well, what if I add in a second trait? Would that impact how that second trait is inherited? In other words, would the round gene impact the color of the pea seed as well? So then he looked at that as well. And this is where things get tricky. In this gamete, we already have the big R from being round, and now we're going to add in the big Y. And all this plant has to offer is that big Y. And in this plant, into the gamete, we're going to add in the little y. It's important that you know in the gametes, I have one R and I have one Y. One R, one Y. Then when these come together to fertilize, this R would come, this little r would come, big Y, little y. To be honest, one of the trickiest things with dihybrids is figuring out the gametes. So we're going to practice this for a little bit and make sure we have this down, because if you get the gametes wrong, then, well, the rest of the Punnett square is really difficult. So our rule is going to be one of each letter and a gamete, okay? All right. So starting with my A's, all right, I have my big A, and I'm going to take that to my first B. All right, and I'm going to stay with my big A. I'm going to take it to my second B. All right, now I'm going to move to my second A. And take that to my first B. Staying with my second A, and take that to my second B. those are my possible gametes. Now, if you want to save yourself some time, at this point you could ask yourself, do I have any repeats? Well, I have 2 with big A, big B, so I could cross one of those. And I have 2 with a little a, big B, so I could cross one of those out too. You don't have to do this, but it may save you some time in the long run. Okay, you pause the video and you do this one. See if you get it right. Okay, let's go over this. All right, so I'm going to start with my R's. And remember, 1 R per gamete. So I'll start with my first R. Write that down. Take it to my first T. T. Okay. To my first R again. Take it to my second T. All right, I'm done with my first R's. Let's go to my second R. Take it to my first T. My second R, and take it to my last T. All right, do I have any repeats? Nope, I have 4 very different gametes. All right, you should pause the video and do this one on your own. Make sure that you get them right by watching how I do it. But please try to do it on your own first, okay? All right, so I'm going to start with letter A. You should have paused the video and already done them. All right, I'm going to start with my first A. And take it to my first B. All right. Is there any other possible gamete I can make in this one? No. All I can do is big A and big B. If you don't believe me, let's go ahead and give it a shot. Take it to my second, big B. All right, I'm done with my first A. Now I'm going to move to my second A. So big A to big B. Big A to big B. All right, so really I just have the one type of gamete that this guy can make. Okay, B. Let's do letter B, starting with my first A. Take it to my first B. Staying with my first A. Take it to my second B. Okay, done with my first A. Now I'm going to go to my second A, so my little a. Take it to the first B. Little a. Take it to the second B. All right, do I have any repeats? I don't. I have 4 very different gametes. Okay, let's look at letter C. Okay, once again, I'm going to start with my first A. Take it to my first B. All right, staying with my first A, I will take it to my second B. All right, I've done all I can do with my first A, so now I'm going to move to my second A. Take it to my first B. And then my second A to my second B. All right, do I have any repeats? I sure do. So I only have 2 different kinds of gametes. All right, let's look at letter D. Starting with my first F. Take it to my first G. And then my first F to my second G. Okay, I've done everything I can with my first F. Now I'm going to start with my second F to my first G, and then my second F to my second G. All right, do I have any repeats? I don't. I have 4 very different gametes here. All right, letter E. Starting with my first F to my first G, my first F to my second G. All right, second F to my first G, and second F to my second G. Okay, do I have any repeats? I do. I'm only looking at these 2. Okay, if you are unclear on that, what I would like you to do is pause the video erase your note sheet, try it again, and then rewatch the video. It's really important you got down how to get the gametes, otherwise moving on to the next step will be really confusing. This concludes our first video on dihybrids. Don't forget to watch the second video.